Hello? Hello? Can you hear me?
Why do you look troubled?
Why is it the screen?
Okay, so let's just go back here to activate the screen, hopefully. And center projector, laptop. And is the projector on? Yeah, it's there. I don't know why the screen is like that, but it just
Okay, wait, I have an idea.
is.
Do you see it? Whoops. Hello?
It works for now.
Great. I'm gonna go listen to this. It actually just
Okay, hi everyone. Hope you're doing okay. It's a really tough time of the year, mainly because we're almost at the end of the semester. We only have about 2 weeks, including this week. So it's really hard to keep focused. It's really hard to make sure that you're doing okay. It's really hard to actually show up to class. So how are you all doing? That good, eh? Okay, awesome. Well, happy Monday. We'll get into what we need to cover today, so we'll just dive right into it. So what I'm going to try to convince you of today is that even though we don't understand the mechanisms by which, after your brain has been injured, and we're using this model of stroke, that after brain injury, your brain actually tries to heal itself. We don't understand all that mechanism. We don't know what's really going on, and there's a lot of hope about stem cells and what they could potentially do. And that makes it really interesting for us, but it also makes it somewhat confusing for us as well. So today we're going to talk and go back over some of the other progenitor cells, why they're useful or potentially useful for us. We're going to be talking about these different types of niches where we have different types of progenitors that are dividing, ready to go at any moment in time. And then how we can actually go back and examine if these are actually helping, not doing anything, or actually hurting our brain after injury. So we'll go over all of those different things today. I think the biggest take-home message from today is that stem cells are going to be really useful to us, but we still don't understand how they actually work. And so the other big technology piece, so if you're in cell and systems biology or if you're a molecular biologist, how do you actually target and kill specific groups of cells? Because one theme that we've had consistently throughout this course is how do you make sure that you're targeting specific groups of cells? And how do you kill only specific groups of cells? We've learned optogenetics, chemogenetics, how to turn on specific groups of cells. But how do we kill specific groups of cells? And today we'll be covering a little bit of that, and we'll be talking about that in the context of stroke. So again, there's going to be a lot for you to know, and today is actually a pretty heavy lecture. There's a lot. This is not a great time of year for us to kind of go into heavy material, but we need to talk about this material, and some of it is actually a review of what we've covered previously in other parts of the course. So I want you to kind of take a good gauge of where you're at right now in terms of your preparedness for the final exam. And again, there are a lot of different genes and proteins, and I'm going to try to make some sense out of them as we go through them. So in the last lecture, and we're going to be covering this from a number of different angles and a number of different ways when we go through the next few slides, and then when we go through the paper as we normally do, there are a number of different niches or a number of different places where in the adult, in you right now, we have stem cells that are proliferating, dividing, doing what they normally should do. And here is an example of the rodent brain where we know that this is happening. And we know that there's the subventricular zone denoted for you on the left as SBZ. That's one place where we have these adult stem cells continually proliferating, dividing, and then they will migrate up into the olfactory bulb where they will differentiate into interneurons in the olfactory bulb. And they actually migrate along a specific pathway, which means that in the brain, if you were to map it out, you could actually see a pathway, just like if you left today's lecture. There are clear pathways up to the back of the room, and this is represented as the rostral migratory stream, or the RMS, which means that there's a pathway for these cells when they divide to get to the olfactory bulb, which is what you're seeing there. The other place where we know that in the adult we get stem cells dividing and progenitor cells continually being renewed is the subgranular zone layer of the dentate gyrus. And these are neurons. These are eventually slated or destined to become neurons, so they might be useful in our conversations about how neurons die after stroke. But then we also have all of these other cells in the parenchyma within the neuronal tissue of the brain. And that means that we have glial cells, which undergo constitutive gliogenesis. They're always proliferating in the background in almost every different part of the brain that you look at, which is what is being shown here. And these parenchymal cells, we can identify them and separate them out from the dividing neuronal cells by specific markers. So we have progenitor cells that are going to be positive for markers like NG2. They will be NG2 positive. We've already talked about pericytes previously in this course as well. These are another subtype. We will have a number of S100 beta astrocytes. So those are markers for astrocytes that have this progenitor gliogenic background. And we've already looked at oligodendrocyte progenitor cells, or OPCs, which will eventually form those different oligodendrocytes that we talked about when we went through multiple sclerosis. Regardless, when your brain has an insult, so going back to what we talked about with stroke, when there is some kind of damage to your brain, all of these cells immediately jump into action to kind of indicate that there's something going on. We will see more of these cells, we will see cells migrating into the area, and they will also start to upregulate and divide more than they normally would. So this suggests that your brain is actually trying to help itself out, and yet in the end, we know that when you look at stroke, that there's actually very little benefit from all of this kind of going on. So what's the purpose and what's the use of this particular upregulation where we get this reactive neurogenesis or we get this reactive gliogenesis? If your brain ultimately at the end of stroke actually isn't getting any better, why bother doing all of this? And is it actually all that helpful, or is it actually keeping your brain from repairing itself? And that's sort of the big question. So we know that brain injury, things like stroke, will actually stimulate neurogenesis, particularly in the mammalian brain, in those different areas, and it might be involved in brain repair. And we think and we hope that these will actually migrate to the area. So you get damage to an area of the brain, we get an upregulation in those areas that I've talked about, like the subventricular zone layer or the subgranular zone layer, where we know that there's adult neurogenesis. They start dividing even more frequently, and they might migrate just like they do through the rostral migratory zone, or stream. They might migrate to the area where they might be needed, or there might be some damage or loss of neurons, and they might be able to mature there. So this suggests, because we get this upregulation enhanced by injury, that maybe there's a route for us therapeutically, because we looked at previously in this course, that there are very, very few, in fact, only 2 recognized therapeutic approaches to different types of stroke. One is surgical, the other one is a clot buster like TPA. We have very, very little therapeutic interventions that can help us in things like stroke. And we know by 2030, which by the way is not that far off, it's literally about 11 years off, that we're going to see a dramatic rise in the number of individuals that are facing stroke. And we have no therapeutic approaches to help us with this increasing sort of disorder that many of you will be encountering, whether it's through your family members or relatives, or sadly, and we don't know all of the reasons why, individuals who are younger are also developing strokes at younger and younger ages as well. So it used to be a disorder between the ages of like 60 and above, but now we see younger and younger individuals actually developing stroke and dying from those different types of stroke. So is there a way for us to be able to look at the effect of neurogenesis? So is it actually useful? And this last bullet point is really the crux of what I'm going to try to convince you of today, that in fact it is useful. And ultimately, we need this to happen, and it does help the brain, but it's not probably in the way that you think that neurogenesis is actually helping the brain after stroke. So they might migrate out of the anterior region of the subventricular zone layer, and we might see more of those proliferating neural stem cells coming from the dentate gyrus, but in fact, they might actually not be doing what you're imagining these stem cells are doing, even though they're migrating to that area. So keep that in mind, because that's really the take-home message from what we're going to be talking about here today. We don't know a lot about stem cells, but we do know that they help, but it's not in the way that we originally thought these stem cells would help. In the last lecture, we also talked about the different types of stem cells that we could find in these different niches, particularly in the subventricular zone layer, and they all have unique properties that are associated with them. Some divide more slowly, and some divide a little bit more quickly, and they proliferate more. And today, that's what we're going to be focusing on. And you'll hopefully understand why this next slide even though it's really, really busy, is an important one in our discussion. Now, there are a lot of proteins, and the first slide that I showed you actually said that by the end of today's lecture, we will have covered over 50 different proteins, genes, etc. And not all of the ones that are shown here are going to be important, but I do want you to know the ones that I think are the most important for today's discussion. So are we okay with that? And are we all on the same page? We're good? Okay, so keeping in mind we have these two different niches. So this vertical line here and this vertical group that you see here represents that subventricular zone. Now, why are we talking about the subventricular zone? This is an area that upregulates neurogenesis after injury. So does the subgranular zone layer over here on the right. But if you take a look, there are some subtle differences and there are some differences in the marker proteins. But let's start here at the very bottom. So here is the subventricular zone. And as they leave the subventricular zone and as they normally migrate up to the olfactory bulb shown here as OB or this purple zone here, they will start to change the expression of the genes, and they will change the types of proteins that they are expressing. So again, surprisingly, and you need to keep this really clear, when they are these progenitor stem cell-like cells that can differentiate into all sorts of things, these type B neural stem cells that we talked about earlier, they're actually positive for GFAP. Now think a little bit about that. You have learned about GFP. In fact, I asked you to know what GFP is. GFAP is this glial fibrillary acidic protein. Normally, you associate that with astrocytes. But what I'm telling you now is that this is also a marker or a gene that is expressed in early development, especially in those dividing, proliferating cells that might become neurons or the neural stem cell lineage. And so we see GFAP positive cells here. The other thing that you should know, because we have talked about this before, these neural stem cells, the ones that could potentially become neurons, also are positive for things like nestin. Not surprisingly, they're also positive for things like SOX2, and these are sort of the beginning, the progenitor cells that we are used to thinking about as neural stem cells. Now, one of the things that happens is as they start to leave this niche where they are used to being in this undifferentiated state, as they stay in this potential to become all these other types of cells or all these other types of neurons, as they leave and start to become a little bit more differentiated, these genes actually start to turn off. And then we will start to see these type C cells, the transient amplifying type, express other types of proteins like DLX2. Now, I need you to be clear on DLX2. There was a lot of confusion last year because DLX2 kind of sounds like double cortin, which we're going to talk about a little later. But DLX2 is actually distal less homeobox 2, that gene, and it's written for you up here at the top if you haven't encountered it before. And DLX2 is actually indicative or is a marker for this type of transient amplifying type C progenitor cell. And again, it's unique and specific to this type of cell here. As it continues to now migrate, and now it's on the way up to the olfactory bulb, we're going to start to see that it expresses other types of genetic markers. But the ones that I'm particularly interested in you kind of knowing is the one here, which is DCX, or double cortin. And this double cortin molecule is a characteristic feature of migrating neuroblasts. Now, I want you to, again, think a little bit about why we might be interested in all of this. Why do we bother with all of these different types of proteins and these markers? Because the key question that I asked you at the bottom of the last page, or the page previous to this, is how do we know that those migrating cells to the area after damage are actually going to be any use to us? So here we are looking at these different genes that are expressed by these different subpopulations of these cells, and these are all derived from those rapidly dividing progenitor cells. Eventually, we want them to form mature neurons. We can mark that with a general neuronal marker known as nu-N. And again, we see almost the same pattern, slightly different, but overall the same sort of markers that we see over here in the dentate gyrus, where they will start migrating from the granule cell layer, and they will incorporate into other parts of the hippocampus. So they will also express GFAP when they're undifferentiated and they're fully considered as progenitors, and they also express nestin and that SOX2 protein as is shown here. And later they will also start to express this DCX, or this distal less homeobox 2, as they start to differentiate and migrate. And again, it's a marker for migrating cells, migrating progenitor cells that are not yet neurons, but could be when they actually arrive in the region that they are going to. And eventually they will form these fully differentiated neurons that we can then mark with nu-n. So the hope behind all of this is that we know after stroke, there's a lot of these cells being produced. We get an increase in the number of GFAP, SOX2 positive cells in these different areas of the brain. And we also know they will start migrating to these different areas that have been damaged. And we know that some of them will become neurons, but ultimately at the end, when we look, there's no functional recovery. They don't actually do all that much better. So can we see what the effect is behind all of this? So again, we're going to be taking a look at some of these different types of cells, and these ischemic stroke models that you should be aware of, being able to discuss them on your short answer questions, whether they're the thromboembolic type that you generate by injecting in chemicals like endothelin-1, or you are doing a middle cerebral artery occlusion where you're blocking the middle cerebral artery either with a little filament fiber or you're clipping it and stopping blood flow. Either way, there's a loss of blood flow, and you're going to see some cells like the type A and C actually start to increase in their number, and they will undergo this mitotic activity within these regions after stroke. And particularly 7 to 10 days after ischemic stroke, we see this increase in mitosis in these areas. There's more cell division, and we know that there is a signal from the damaged area that actually goes on to signal for increases in neurogenesis. So this subventricular zone region might actually be a really important one for us to think about therapeutically, since we don't have any of those therapeutic interventions that we want in medicine, and particularly with a biological background. So here's why we're interested in all of this, the big picture. Really smart people have been looking at what's going on, including those here in Toronto. So if you know Dr. Mooreshead and you know other individuals who've been working on neurogenesis, stem cells, and other things, people have been thinking about this, worried about this, considering this, and also hypothesizing about what's going on here for a long time. So it's not really clear why this proliferation occurs. That's the take-home message that I've been repeating several times in today's lecture. And we don't really know what might happen if this proliferation doesn't occur. And that's going to be the topic of what we're going to cover here today. And again, people have used these models, and I'm going to take you through these models where really bright individuals, not just here in Toronto, but in the United States, in Europe, and generally all over the world, have been looking at what happens after stroke, what goes on with this proliferation, and what are some of the confounds. Why don't we think that this might actually be useful? So what we're going to be looking at is how do we specifically target a class or a subclass of cells, and in this case, you've already got the punchline, which is double cortin-containing cells. Why double cortin? Go back to that previous slide. These are those migrating neuroblasts. They're the ones that might differentiate into neurons. They're not quite progenitor cells, because they would be GFAP positive, but they're not quite neurons yet either, and so these cells might be really useful to us. But how do we eliminate this group of cells that might be important in the brain after stroke? And it turns out that we can specifically target any group of cells that we want as long as we know what genes are specifically expressed within those different groups of cells. So again, to give you a little bit of background on all of the different things that have been done in this field up to this paper that I just showed you on that previous slide. So up to the Greenberg paper, people have been thinking about this for a long time, and there are some crude approaches, old approaches that people have used, including ones that I used to be involved with when I was doing graduate studies, which includes just irradiating them. If you do gamma or X-ray irradiation over an area of the brain, those cells die. So as they try to replicate their DNA, they can't because their DNA is actually damaged. And in those brain regions where we have lots and lots of cells dividing, if you X-ray irradiate them, you can actually cause all those dividing cells to die. So people have done that. You can also inject in antimitotic agents or anti-proliferative drugs. So cytosine arabinoside, also called Ara-C, have been used to try to block all of this proliferation after stroke and to examine all these things that happen after stroke. So people have been thinking about this for well over 20 years, and they've taken these crude approaches to this. I've already shown you that GFAP is a marker for those proliferating groups of cells, as well as nestin. And in fact, people have been using this homologue of Drosophilus thales, which is a gene expressed in Drosophila. And what they've been looking at is to express a homologue that actually will express thymidine kinase, which is going to be crucial to our discussion a little bit later. They've been trying to do this in the background of GFAP and in the background of nestin. And if you've been following and you haven't yet fallen asleep and you're still with me up until now, you might be scratching your head going, but then why bother doing that PNAS paper in 2015 if people have done this already? Well, there are some confounds because these studies that have used these genetic approaches are actually targeting an earlier population. Remember, GFAP is expressed in those subventricular zone progenitor cells that could potentially become double cortin positive and other new N positive, but that's really early in the process. And so this ablation, these early ablations where they're targeting nestin or GFAP positive cells actually have a number of different possible interpretations, and therefore they are confounds in our understanding. And some of the other things that have resulted from all of these other types of studies that are confounds and why we need to have a better follow-up study are things like if you do this type of ablation where you're killing off groups of cells in large numbers using X-ray radiation, or you're using cytosine arabinoside and you're injecting in cytosine arabinoside, you're actually going to invoke an inflammatory response. But guess what? The inflammatory response, if you are turning on inflammation, might also impact how you can look at the results. So we don't want that. We want to be able to kill off these cells without having a large inflammatory reaction after the delivery of drugs like cytosine arabinoside or other anti-proliferative drugs. We might also be recruiting, as a result, microglial cells, which are normally not part of the nervous system. The microglial cells embryologically actually come from the yolk sac. They're not derived from the neuroectodermal cells. They have a different embryological origin. And so these microglial cells might actually also be contributing. And by doing these different types of antimitotic, anti-proliferative drug delivery, we might actually be changing the blood vessel anatomy or the circuitry of these blood vessels, which again might not be something we want. So there are a number of these confounds, including GFAP, I told you earlier, and this is this last bullet point. So if I only want to target neurons, and I only want to worry about the groups of neurons that have started to divide and are migrating to the area, and I want to see their contribution, GFAP also exists within astrocytes. GFAP also exists within those glial progenitors. So how do I know that those glial progenitors, these non-neuronal cell lineages, are not going to be involved? So I might use GFAP, I might target them, but in fact, I might be making another confound directly by how I create sort of the transgenic approach. So I'm hoping that all of those confounds are kind of clear to you. I'm hoping that you understand why this paper might potentially be interesting for us to study, but it's also a case study for you to make sure that you understand the different approaches and why specificity in science is incredibly important, because it addresses many of these different issues that are shown here. So again, this is sort of a background slide for you, and if you already have most of what I've been telling you, then you don't really have to memorize this, and this will be repeated several different times. So this is a sort of low-level view of the brain of a rodent. And what you can see here is the olfactory bulb. The cortex is here at the top. And this represents the rostral migratory stream, or the RMS. We know that this is connected to the subventricular zone. There is a direct path, just like there is a path available for me from here at the front to the back with the stairwell. There's a pathway that these dividing neuronal progenitors can actually follow to get from here on the right to the olfactory bulb on the left. And we can actually stain the neurons that are migrating along this pathway because these migrating neurons are positive for double cortin. Right? So these migrating neurons, the ones that have left, they're not neurons yet, but they could become neurons. These migrating neurons are actually staining positive, this green that you are seeing here, along this rostral migratory stream. So these are examples of how we can actually tag or go back and see these migrating neurons that we can mark with double cortin, which is a marker for migrating neurons. Are we good? We're not good. Are we somewhat good? Thank you. Okay. So if we're somewhat good, let's just keep going. So the other thing that I can do, and I'm going to come back to this again a little bit later, I want to know if, in fact, these double cortin cells, because, hey, they could have come from anywhere. I don't necessarily have to believe what I am learning here in CJH, nor do I have to believe what I see in other papers. Are these actually coming from dividing cells? So one of the things that we can do is inject the animal before we do this type of study with a marker known as BRDU, or bromodioxyuridine. It's a synthetic analog of uridine, and we can detect that as cells divide, they incorporate BRDU, so dividing cells will incorporate BRDU, and any cell that has divided after taking up BRDU, we can still track it. And in fact, when we do so, we see BRDU staining in the same area within the RMS. So these dividing cells that started off in the subventricular zone are actually found in the same places that we see double cortin. So BRDU does not normally exist. We have to inject it. It's very, very specific for any dividing cell that has taken up BRDU. And we can see that for the most part, when we look, any cell that is double cortin or a migrating neuroblast is also positive for BRDU in this rostral migratory stream that you are seeing here. And in fact, a higher power view of one single double cortin positive migrating neuroblast. So here is a putative dendril, and then you can see in the middle the BRDU in the nucleus. Here is a high power view that yes, this migrating neuroblast actually has BRDU within the nucleus, just as we would suppose. So this again seems to suggest double cortin is a great marker for these migrating neuroblasts that might become neurons when they reach their target that they're hoping to get to. And we know that they're not nu-N positive. So they're not neurons yet because nu-N is a marker for fully differentiated neurons. And we see very, very few of them in this area of the brain that are positive for both BRDU as well as the differentiated marker nu-N. But we do see some of them in the olfactory bulb, which is shown here in E. So some of them did make it and did become fully differentiated neurons after they have been tagged. Any questions on this before we go on? Seeing as there are none, we'll just continue on then. If you have questions, please let me know. So we know, again, going back to the beginning, we know that injury actually enhances neurogenesis, and this might be involved in brain repair. We don't know if it's actually involved in recovery. There is no, you can't spontaneously by yourself after a really traumatic stroke recover fully. If it's been a bad stroke, again, think anecdotally, you're likely not going to get better. You need some intervention, etc. But we don't know what the role in recovery is. That's what we're trying to find out through these experiments. So we could go in and we could try to prevent this neurogenesis, and that has been done in the past. And in the past, people have tried to come up with an explanation for why neurogenesis might be important, but then there are all these confounds, like when we do these anti-proliferative drugs, or when we knock out GFAP positive progenitor cells, we are losing populations of glial cells, or we are inducing an inflammatory response, etc. So could we get a better, more specific group of a specific group of progenitor cells that could tell us that this migration to an area after damage is actually useful? So we have one, we have a putative candidate, and this is double cortin. So double cortin is a microtubule associated protein. It's part of the sort of microtubule network, and it's an adjunct protein. And this is found in newborn neurons, neurons that are just coming out of that proliferation. And more importantly, they're also part of these migrating neurons. So here, it's a little bit better than the types of experiments that have knocked out GFAP positive, nestin positive, because go all the way back to the first series of slides, nestin, GFAP, they're great for the progenitor cells, but they're not quite at the level that we want, and GFAP is also found within those glial cells as well. So knocking out glial cells might just be equally important to knocking out migrating neurons. So the authors of this current paper are trying to address this. So in the past, there have been all of these different types of confounds. So let's go back and try to express this heterologous, so it's normally not found in the mammalian system, this thymidine kinase that's normally part of the Drosophila pathway. Let's try to express this now in specific groups of cells, and particularly under the control of double cortin, the promoter for double cortin. And we've talked about why knowing promoters and how we can use promoters to drive the expression of specific types of heterologous systems, like channel rhodopsin is a good example. We could put channel rhodopsin in specific groups of GABAergic neurons, using things like calbindin, or we could put them in glutamatergic neurons by using the VGlut2 promoter, etc. So here, let's put thymidine kinase under the control of this double cortin promoter, which I've tried to show you is involved in newborn neurons that will migrate to an area of damage, or potentially of places like damage following stroke. So again, could we go in and take a look at this, because this is better, and it's a better marker of the neuronal lineage than GFAP. That's really too early. And GFAP is also found in glial cells, and so we might be knocking out glial cells at the same time. And nestin, again, is an early, very, very early neuronal progenitor marker, but they're not the ones that will actually leave the target tissue. So this candidate gene, double cortin, might be of use to us. So do you have any questions now? Yes? So they haven't differentiated into neurons. They will when they reach their target, normally the olfactory bulb, or if in the area of stroke, like if they reach and the environment is suitable, they will differentiate into neurons. But neurons themselves can't migrate.
So what happens if they don't differentiate them? They just die?
So if they don't differentiate, so if you're talking about, so let's look under normal conditions. So if we look along the rostral migratory stream and they're not reaching their target, what would happen to them? And in fact, they continue to actually search for their target. They're not in a differentiated state, and as long as they're getting local support in the rostral migratory stream, they'll actually be able to survive. Any other questions? No. Okay. So again, some of these details are really not that important. You don't need to know how many kilobases upstream of the promoter that we're actually looking at. But if we were to take a look at how they actually did this, so they actually are doing things a little bit differently than we've been looking at previously in this course. Previously, we've been looking at the delivery of different genes through viruses. We package them into viruses, and then we deliver it into a mouse. Here, we're creating a transgenic mouse line. And again, I don't need you to know all the details of these transgenic mouse lines. If you're really interested, the details are up here for you. So one of the things that they do is they take part of the double cortin promoter, and then they will actually use this to create these transgenic, and these transgenic mice will have kinase in any cell that contains double cortin. And the second bullet point is actually there to show you how they did it. So how do you take this gene and how do you fuse it with thymidine kinase, and then how do you transfect embryonic fibroblasts and then create these transgenic lines from this? And again, all of those details are really not important. Suffice it to say, we need to keep some of these in mind because we already have talked about glial fibrillary acidic protein, or GFAP. These are also astrocyte markers. You know about oligodendrocytic markers, so GAL-C is one that we could use to check, are we affecting oligodendrocytes? Are we affecting neurons? Are we affecting, for example, astrocytes? And it turns out that when you create these double cortin thymidine kinase containing transgenic mice and you breed them to the first generation, or F1, again, the details not really important, that we do find thymidine kinase, which I have not talked about at all other than one line early on. Thymidine kinase, normally not part of the mammalian system, is actually found within any cells that contain double cortin. And that's, again, the most important part to this. Double cortin promoters can be used to drive the expression of thymidine kinase. And these mice that result from these transgenics appear really healthy. They have normal brain growth and development. Their reproduction is not affected. And their double cortin levels are not affected. So they look like any other mouse. So by creating these transgenic mice that now contain thymidine kinase, there's no difference. They look like normal mice, except they're carrying thymidine kinase in any cell that also has double cortin. Good, not good, somewhat good? Good, okay. So, all right. This part I do need you to understand, and this is actually critical in how we're going to approach what you need to know for the final exam as well. So, how do you kill specific groups of cells? So keep in mind, I haven't talked to you about thymidine kinase, and this is what this slide is actually showing you. Thymidine kinase is actually not part of our genome, so we have to introduce it. And if we have it, does it really affect our genome? The mice actually suggest it doesn't. But what does thymidine kinase actually do? So thymidine kinase is really not going to do very much because normally it's a virally encoded, and it could be found within different genomes, but it's a virally encoded kinase that can convert ganciclovir and put a phosphate group on it. And again, ganciclovir is an antibiotic. It's normally not something that we encounter. In fact, ganciclovir is not really readily prescribed anymore for any normal type of condition. So thymidine kinase we normally don't find, nor do you normally encounter ganciclovir. The mouse cannot go to the mouse pharmacy and ask for a prescription of ganciclovir, so the mouse is not going to encounter ganciclovir. But here's why it's an important consideration for us. We don't normally have it. These mice now, in every cell that contains double cortin, now has this exogenously, heterologously expressed thymidine kinase. So what's the problem with this thymidine kinase? In the presence of a drug or antibiotic like ganciclovir, this thymidine kinase that is normally not part of the mouse's genome will put a little phosphate group on ganciclovir. This actually prevents it from leaving the cell. So it enters a double cortin positive cell. The double cortin positive cell has thymidine kinase, and it actually traps it, just like when you are starting to look at Krebs cycle. Once you phosphorylate something, it can't actually leave the cell anymore. So now we have a little phosphate group. And then other cellular kinases found inside the cell are now capable. They wouldn't have been able to previously, but now they're capable of adding a second phosphate group. So without that first phosphate group, intracellular kinases cannot do this at all. It requires that first phosphate group that's coming from thymidine kinase. And then once it does that, it will actually add a third phosphate group. So that first phosphate is actually critical to this entire conversation. And these host cell kinases are all intracellular. They require that first region to be phosphorylated first, and then through steric modulation, the second, and then the third phosphate group to this. And this triphosphate group that you see here on the far right is actually toxic to the cell. So again, keeping in mind, this whole process can only start in a cell that has thymidine kinase. The only cells that have thymidine kinase are double cortin positive. And so why is this bad? This compound, as it builds up, is actually an analog, a purine analog, that actually inhibit DNA polymerase. And as this builds up within the cell, and as the cell wants to be able to create new DNA, it will actually cause this cell to die. And these cells will die through apoptosis. This is also useful, by the way, and you don't need to know this bottom part about the bystander effect. This can actually be used to kill other nearby cells as well, especially in things like cancer. I think my microphone went out, but I think everything else is still good. And so this means that if we're taking a look, this gives us a very, very specific way to target thymidine kinase to a specific promoter-driven group of cells. And this in turn means that we can, by adding ganciclovir, kill that specific group of cells. So that whole pathway, that whole specificity of killing groups of cells is actually a really important one that you should be able to take away from today's lecture. And if you can't, and you don't understand how this works, then it's something that you should come to see me about sooner rather than later. You will have to demonstrate, not by repeating back what I've just said, but be able to apply it in targeting specific groups of cells. So I might ask you to design an experiment where you are able to demonstrate that you understand this as well as specific promoters by telling me how you might kill off a group of cells that are inhibitory. And you might be thinking of how you might do that right now. And again, I need you to understand how this whole process actually works. So again, the structures that are on here for the ganciclovir, you're not going to be asked to draw them. You're not going to be asked to go through all of these bystander effects down here at the bottom. You might hear of them in other courses because it's very useful in killing off cancer cells as well. So does this actually work? So does this process actually work? And the next few slides are actually just showing you a proof of principle that these transgenic mice that now have thymidine kinase are actually normal. I've already shown you that earlier. I've already told you that earlier, but that was just a bullet point. And what all of these slides are now showing you here is that double cortin, the expression of double cortin in the double cortin transgenic mouse versus the wild type, the levels of double cortin are actually the same as we would expect. There's no increase in the intensity of the Western blot. So by creating a transgenic where the promoter is being expressed at high levels, it actually doesn't affect double cortin levels. If we do staining for double cortin and we look at the subventricular zone layer and we look for these little brown spots, do we see double cortin both in the subventricular zone layer, which we do, and do we also see them as these migrating cells that are leaving the subventricular zone layer that will eventually hit the rostral migratory stream, and these little brown dots that are stained with immunoperoxidase labeling actually show you the same thing. Do we see the same type of pattern in double cortin transgenic mice? We also see exactly the same thing going on here as well. And if we take a look only in the double cortin transgenic mice that contain thymidine kinase, we can see thymidine kinase in green staining in cells that are double cortin positive. And only the double cortin positive cells are the ones that actually have this green staining. We don't see green staining without the red double cortin staining. So it is very, very specific. And this is really just showing you that double cortin is not affecting normal development, and it is targeting the place that we would normally expect double cortin to be found. So can we actually show that in the presence of ganciclovir, which is the compound that would normally cause the inhibition, or sorry, cause the loss of these cells in the presence of thymidine kinase, and we have just created these thymidine kinase positive mice, can we actually show that ganciclovir can wipe out this population of these neuroblasts? And it turns out that yes, we can. So this is, again, wild type. And if you give wild type animals ganciclovir where they don't have thymidine kinase, it doesn't affect double cortin staining because those neurons don't have thymidine kinase. They cannot put on that first phosphate group. They don't cause the apoptotic loss of these cells. But if you do the same experiment in this double cortin thymidine kinase transgenic mouse, and now you give it ganciclovir, and you go back, do we have double cortin positive cells? In fact, we will wipe them out. We don't see them in the subventricular zone layer. There might be a remnant of one here at the arrowhead, nor do we see them in the rostral migratory stream as well. So we will also wipe out the whole population of double cortin proliferating cells after labeling them with BRDU. This is normal mouse here, and we can see BRDU is also being affected. So again, all of these things suggest that in these different mice, after the addition of ganciclovir, that we have a selective loss of double cortin positive migrating neuroblasts. Are we all good on that? Do you believe this data? Data looks really convincing. If you were reviewer number 2, you would say, go for it, publish it, it's good. You guys are tough reviewer number 2s. Yes, you have a question? I'm not sure what I said in the last little bit. Is there, yep? So if we take a look at the section similar to what we saw before, and we're staining for double cortin, we're no longer seeing double cortin, and we are also seeing that these double cortin cells, which are normally part of the division process, so that's still fairly early, but it's not at the very early, the earliest possible stage, we're actually seeing a loss of proliferation markers like BRDU if we injected them. So this is sort of all background to what we actually want to know. So what is the purpose of this dividing population of cells which actually get increased after stroke? And so many labs have actually shown this, that it's actually worthwhile going in and investigating. So what happens if we actually wipe out this population after things like MCAO, which is that middle cerebral artery occlusion? So if we block the blood flow to an area of the brain that is involved in motor movement, and this area of the brain is known as the striatum, and I'll show you this in a moment, what happens to the animal functionally and at the cellular level if neurogenesis is no longer happening, because I can wipe out those specific groups of migrating cells after damage? If that's not taking place, what do you think will happen to that lesioned, stroked out area after I have this middle cerebral artery occlusion? And that's sort of what we're going to be looking at. I think before we get there, though, we should probably take a break. Your synapses kind of look like they're a little bit frazzled right now, a little bit too full. And so taking a little bit of a break now might be the best idea. So we'll come back in 10 minutes, and then we'll continue on to see if this actually has an effect.
so sorry. I'm so sorry. Hi, my name is Sarah Bello. I'm the third year rep for CSBSU, and we're having a Meet Your Profs night that I thought you guys might be interested in. I think that you might be interested in it because one, you get to network with professors, and you might get like research experiences, right? So that's always great. And also it's free food, and it's pickle barrel food, like not pizza, but like actual food, right? So I think you guys should come. It's today. I know short notice. Sorry. It's at our, let me get the room right. RW 432, and it's today 5 to 7. So hopefully I'll see you guys there. All right. Thank you.
All right, if you don't mind, I think we're going to get going on, and I'm going to go over panel E with you in the next few minutes. And one of the things that you need to be really good about for your final exam is when you're asked about ensuring that it is specific. It's not only about if we're knocking out a specific group of cells. It's not just to demonstrate that yes, the double Corton positive cells are the only ones that are proliferating, they're the only ones that contain thymidine kinase, but equally importantly, the other populations of cells that could be affected, the ones that are GFAP positive, are not changing in number, nor are some of the other nearby cells. So for example, after ganciclovir staining, we don't see any changes in GFAP levels. I didn't show you this here directly, but galactocerebroside C, or a marker for oligodendrocytes, that's not affected. So all of the other cell types that we might be interested in that come from different progenitor lineages are not affected. It's only the cells that have double Corton. And in addition to that, when we look at staining for microglial cells, so the antibody itself is called OX42. It stains for a marker known as IBA1. There's no upregulation of IBA1 after double Corton has double Corton and thymidine kinase in the presence of ganciclovir have wiped out that population of neuroblasts. So we don't get this upregulation of the immune response, and this meets and it checks off each of the boxes that I showed you before. There's no change in the immune status, there's no inflammation, none of the other cells are being affected, and it seems to be very, very effective in only wiping out the double Corton cells. So it is very specific. And these are all sort of measures that you need to be able to address. Other cell types are not affected, and think about that on your final exam for showing specificity. It's not just the presence of a change, but it's also the absence of changes in the other populations of cells as well. So going over this, and again, the time points, not so important here, but we're going to come back and talk about it on a sort of big picture level. So we do this focal cerebral ischemia here by using middle cerebral artery occlusion, or MCAO. We do this on these double Corton mice, which I've shown you up until this point in time are perfectly normal. They can have ablation of double Corton positive cells, and then we are going to treat them afterwards with either saline or with ganciclovir, which, if it does its job, which up until now it has been, will wipe out that population of migrating cells after stroke has actually occurred. And again, they are also going to undergo sensory motor testing to see if there are functional deficits that can be compensated for or are lost after ganciclovir training. And some of the different types of testing that are done include the rotarod test for motor performance, limb placing. So if you create a stroke on one area, do you affect the other side of the body? Does it use both hands equally? And that's known as limb placing. And then elevated body swing tests. If you're affecting one side and you stroke one side of the body, it's really hard to control the muscles to swing to the opposite side. So do you see that there's a loss in swinging if you're held by your tail to the opposite side? In these ganciclovir treated mice, where after stroke, we don't get that migration. We don't get the neurogenesis that we would originally have thought. And again, here's an example of what the rotarod test looks like. They're put on this rotating rod. And we want to know, after stroke and after treatment with either saline or with ganciclovir, where we're wiping out those migrating neuroblasts, how is their motor performance actually affected? And it's a really simple sort of measure. We just see how long can they stay on this rotating rod. And the take-home message from all of this is that if you give a wild type a stroke and you give them PBS, or you give them ganciclovir, again, there is no thymidine kinase in these wild type mice, that this is sort of the normal time that they're on the rotarod. They have been affected. They're not staying on as long as you would normally expect with an unstroked mouse. But after you have the double Corton thymidine kinase and you have killed those cells with ganciclovir treatment, you can see their motor performance. They can't be on the rod for very long. You have affected their motor performance and recovery after stroke by eliminating those migrating neuroblasts. The placing score, so how often do they use one side versus the other? And the higher the score is, the more often they are using both the left and right equally. But with ganciclovir, if you stroke one side of the body, they don't get that recovery or the recovery back to the same baseline level. So ganciclovir, again, causing in these double cortin thymidine kinase expressing mice very severe, pronounced motor effects that seem to be quite impactful. And then also the left body swing, they're not able to move their body equally because their motor, the caudate putamen is actually affected more in these mice after stroke. All of these measures, which are functional measures, so very similar to what you would have done if you were going in for physiotherapy, all of these suggest that if you don't have these migrating neuroblasts around, you actually perform and function less well, not unexpectedly, but we'll talk about that in a moment. So if we go and take a look histologically, so let's take a look at the brain sections and see what happens to the brain. Can we also see, not just at a functional level, which was the previous slide, but also on a histological level, can we actually see damage to different areas of the brain, so the caudate putamen, motor cortex, etc., after these different types of stroke. And one of the most useful compounds to be able to do this is actually TTC, which is a form of tetrazolium hydrochloride. And we can apply this to a living tissue. So we take out the brain, and then we would section the brain. That brain tissue is still alive. We do that for electrophysiology, but we can also do this just to see what the tissue is doing under normal circumstances. And then we can see what happens to the dye, this TTC dye, after we have stained this particular brain. And we can compare it to the brain of an animal that has been subjected to stroke versus an animal that has the migrating neuroblasts wiped out in a very specific manner. Is the lesion size going to be bigger, or is it going to be smaller? And so if you take a look, this is sort of what happens. TTC is a sort of white salt that is applied to the cell. So you take the section of the brain, which is what you see here. You incubate that section of the brain with this tetrazolium salt, or TTC, and then you wait. And if the tissue is still alive and intact, this clear, colorless white salt is actually taken by living cells with cellular dehydrogenases, and only living cells will have these cellular dehydrogenases, and the metabolite actually turns red. So wherever you see red, in other words, the cells still have cellular dehydrogenase, which means that the cells are still intact, the cells are actually still alive after stroke has occurred. And one of the things that you will see is that if you just treat a double cortin thymidine kinase mice, give it a stroke, which is everything that you see here in white, those cells have died, that area of the brain has died. This is sort of the loss that you would expect across the motor cortex, across parts of the caudate putamen and parts of the striatum. But if you gave it ganciclovir, the size of the loss that you would see after stroke is actually substantially and significantly larger. So again, maybe not surprising that not only do we see worse performance functionally, the limbs aren't moving as well in these mice after we eliminate migration of these dividing cells, but histologically we also see a bigger infarct, and maybe you're not surprised by that. So this suggests that if we take out these double cortin positive cells, and if we eliminate this migration, because these double cortin positive cells are not just involved in proliferation, but they're also involved in migration, that we are going to affect the performance as well as the amount of cell death that we can measure within the brains of these mice. So again, maybe not surprising for you, but this actually addressed a lot of the previous ideas and these conditions about, is there immune system activation? There is none in this case. So microglia aren't involved. They don't see changes in any of the other cell types. And so this was sort of a better overall model. And again, without going into a lot of detail, into the target areas that were damaged, do they see in the same side that you damaged and you did a lesion after middle cerebral artery occlusion, did you actually see the migration of these neuroblasts to those areas? And if you wipe them out with ganciclovir, did you see migration to these areas? The take-home message is the bullet point at the top. There is no migration after ganciclovir treatment. Therefore, there's no new neurogenesis, and that accounts for a loss in viable cells at the area following stroke. Again, maybe not surprising to all of you when you're thinking a little bit about what this might represent. So this worsening of the outcomes, eliminating this neurogenesis, so we did it in a very specific way, introducing thymidine kinase in double cortin positive, proliferating and migrating groups of cells that we normally see proliferating more after stroke. By doing so, this suggests that this neurogenesis is actually beneficial. Again, maybe not surprising for you. But think a little bit about this, and I mentioned this in passing. So one of the things that I didn't talk too much about is the timeline for these changes. So we're affecting neurogenesis like a week later or 2 weeks later, or we're introducing stem cells, or we're reintroducing stem cells in many of these studies after significant time has gone on. So if we're looking at so why do we lose so many of these neurons? Why is the size of that TTC staining actually so much different? The time frame actually doesn't account for it. Neurogenesis actually takes weeks to actually occur. So from the time it's born to where it migrates, it actually takes a long time. But we're actually able to see this within 7 days. So the timing of this is quite a bit off. And so this actually raises some more questions. So this study in itself, although really helpful, it tells us that without any influence of the inflammatory system, without microglia being involved in a very specific way, taking out a subpopulation of cells, that we are actually showing that neurogenesis is beneficial. The time frame actually suggests that these migrating cells are not replacing the neurons at that area. They're actually doing something else. And so this mechanism other than normal neuronal replacement, so the size of that infarct is bigger or smaller, not because neurons are replacing damaged neurons, but something else is going on. They're not migrating and then taking them over. They're doing something differently. We don't really know what that means. And one of the proposals is based on these types of studies where the timing can't be attributed to new neurons coming into an area of damage, is that maybe these stem cells are actually releasing factors, other factors that might be beneficial to damaged neurons after stroke. So some of these growth factors might be things like vascular endothelial growth factor, or VEGF. So one of the places where we see lots and lots of VEGF is if you go out and exercise, not only do you produce more BDNF, or brain-derived neurotrophic factor that's good for your brain, but you also increase the production of VEGF, which we don't really talk too much about. And this VEGF is actually really important, especially if you're thinking about stroke and the production of new blood vessels to an area of the brain after stroke. Something in those stem cells or migrating neuroblasts might actually be released, and this factor might be VEGF. And this VEGF has actually been really important in studies of ischemia for the growth of new blood vessels. So this kind of makes it interesting. It makes us kind of pause a little bit. It makes us think a little bit about what we think normally happens if we have stem cells and they're migrating to an area. We'd like to think that they are replacing those neurons that are lost through the mechanisms that we talked about last week. But this actually suggests, and there's more and more data to suggest, that maybe it's not that simple. It's not a simple replacement. So this leads into what we might start thinking about next, which is other factors. And these other factors are actually maybe similar and take the same approach, but might be a little different than what you might have been thinking about in terms of stem cells. So again, VEGF has had a number of different effects, not just these new blood vessels. It's also been proposed to be anti-inflammatory. Maybe it's like really beneficial, et cetera. So we're going to come back and talk about that in this context, and then we're going to wrap up this course talking about some of these different issues. And in particular, I'm going to be mentioning Jason Shepard and Vivian Budnick and their contributions to what we think is really an exciting area in neuroscience research when they came out with their papers back-to-back last year. And some of this gets into our conversation now, which is about exosomes and what might happen next in terms of Alzheimer's disease and how we might be able to treat Alzheimer's disease. So we're going to be talking about these different types of extracellular vesicles, these EVs. These might be really important in stroke therapy for both practical reasons as well as for theoretical research-related reasons. And I wanted you to understand why we're interested in this. And these extracellular vesicles might actually be a really interesting way that neurons, other cell types within the brain, actually communicate. And are they just as good as stem cells? And that's really the key. And the findings that came out last year, how do they relate to Alzheimer's disease? And why might they actually be the therapy that cures us of Alzheimer's disease when we start talking about it next week? So when we look at the loss of neurons, particularly in this model that we've been looking at related to stroke, and one of the things that we want to do is to get recovery. That's ultimately the goal. It's not just about developing these different models. We want to actually develop long-term functional recovery as well. And right now, there aren't very many. And I've constantly reinforced that with you over the last little while. That in fact, we only have these two recognized approaches. And one reason why we've gone off into this sort of tangent about stem cells is that stem cells had all this hype associated with recovery after stroke. But yet, when we look, our animal models give us very, very confusing data. And the only two recognized approaches that we're still left with in 2019 is either clot busters like tissue plasminogen activator or mechanical removal of either a hematoma, bleeding out, we remove the hematoma from your brain through neurosurgery, or inside a blood vessel, you would actually extract the thrombus or the embolus directly. And those are the only two recognized approaches still to this date in 2019. There are no drugs that are currently in clinical trials, et cetera. So one promising approach to all of this is really kind of a mix of neural stem cells and pharmaceuticals. And this gets into this other strategy that people are thinking about, which are related to these extracellular vesicles or these EVs. And these are small particles that are continually shed from the surface of all cells. So all cells, not just the ones in the nervous system, and each and every cell in the nervous system, oligodendrocytes, precursor cells, neurons, microglia, every single cell that we look at always is shedding. Just like as I'm walking back and forth across this stage, I'm shedding skin. So wouldn't it be a remarkable thing if somehow I was able to impart knowledge by shedding my skin on all of you? It'd be both gross, but also like really compelling, right? So, you know, what can I say? So this is sort of the novel idea that somehow there's this extra form of communication that can actually be of benefit to us that has up until the last 10 years been invisible to us. We haven't really been thinking about this in the nervous system. We think about neurotransmission, and we think about how action potentials cause the release of neurotransmitters. And it turns out that neurons, glia, progenitor cells, they're always communicating with each other through these extracellular vesicles. And this is actually a very, very strong area of research related to therapies. So you'll be hearing more about extracellular vesicles in about 5 to 10 years when more of these therapeutic approaches actually come to pass. And here's a couple of reasons. And if you went through last year's final exam in the exam repository online, I asked, like, do you remember the reasons why extracellular vesicles are actually beneficial? I can't give you more than 1 dose of a stem cell. So you come in, let's say stem cells work. I can give you like 1 shot of stem cells, but I can't actually give you a second shot of stem cells. That's not currently done. It's not clinically and therapeutically feasible. But these extracellular vesicles that are derived from stem cells, I can give you them as many times as you can take Tylenol. And so we can actually give multiple doses of extracellular vesicles, but we can't with stem cells. You're given 1 dose of stem cells, and that's it. You don't go in for multiple treatments. We can also store these. So imagine one day down the road that you have issues related to a stroke, and let's say we have a therapeutic approach to stroke, and let's say it involves stem cells. If you've ever grown cells in culture in one of your labs, you know it takes a lot of equipment to be able to do that, and you can't easily transport a whole bunch of cell culture related material in the back of your ambulance. But you can store extracellular vesicles because they're very stable under certain conditions. And so you don't also need advanced training. Like I don't have to open up your skull. I don't have to implant them in a specific area. I don't need stereotactic knowledge. If in theory what I'm going to tell you is true, all I have to do is to be able to inject into one of your blood vessels. So again, there's a lot of promising therapy related to these extracellular vesicles that actually might be related to all of the stem cells that we've been talking about. So a number of different stroke publications. If you go back over the last 10 years or so, if you look at some of the ways in which people have tried to address stem cells, it's not just the stem cells that we're looking at. There are a whole host of other possible stem cells that actually are not neuronal in origin. So these are mesenchymal stem cells, which are often abbreviated as MSCs. And these mesenchymal stem cells embryologically have a different sort of background than the neurons or the neuronal stem cells. And if we give the extracellular vesicles that are derived from these mesenchymal stem cells after a stroke, we actually see that they're beneficial. And this is why we're interested in this particular therapeutic approach. So in rodent models of stroke, and you can think of all the different rodent models that you know of how to induce stroke, and every last one of them have been attempted. So whether endothelin-1 injections, or whether you're doing MCAO clipping, or whether you are injecting in thrombin, all of these have worked in these different models. And in fact, the behavioral improvements, so think about all the behaviors you learned about today, body swings, the use of different forelimbs, the rotor rod tests, all of these have shown functional improvement after the delivery of these extracellular vesicles and the functional significant reductions in brain infarct size. So how might I have measured this reduction in infarct volume? How could I have done that after inducing a stroke? I can't tell if you're trying to fly away or if you're, yes. So I could use that tetrazolium salt. So all of these things are now ones that you can imagine how you could do these experiments. So there are a lot of different things that make extracellular vesicles very exciting, but they also make it somewhat confusing because what's entrapped inside these extracellular vesicles, the cargo, what's inside the extracellular vesicles, are actually dependent on the source that they're coming from. So in other words, if I have extracellular vesicles that are coming from oligodendrocytes, they're a little bit different than extracellular vesicles that are coming from neurons, and they're a little bit different than the extracellular vesicles that are coming from mesenchymal stem cells. So I need to know where that source material is because the biological properties of the resulting extracellular vesicles change as a result, which means that we need to compare. So most of the studies that I showed you earlier are actually done using mesenchymal stem cells. They're easier to derive. We have better sources of mesenchymal stem cells, and they're just a little easier to work with in general. But if this second bullet point is true, then these neural stem cells should actually have different extracellular vesicles than the mesenchymal stem cells. And if most of the research has been done using mesenchymal stem cell extracellular vesicles, and that second bullet point up here is true, we should go back and examine the neural stem cells and their extracellular vesicles, which was one of the first papers that was done at the tail end of 2017. So again, these mesenchymal stem cells are these adult stem cells, often coming from a number of different places. One reason why I said that they're actually fairly easy to derive and fairly easy to obtain is that we can actually reprogram adipose tissue. So you can take adipose tissue from an adult fairly easily and reprogram them back into these mesenchymal stem cells. You can get them from bone marrow, et cetera, from the umbilical cord. So we have a number of these different things. And these normally will form things like bone, cartilage, muscle, other fat cells, and connective tissue, but they also are beneficial to the brain. And you have to think why that might be the case. So all of this suggests it might be really, really important. And it shows that maybe if we think a little bit about this, and this was a confound. So here's all this stuff that's normally good for your bone, good for your cartilage, good for things that are outside of your brain. And now when we inject them systemically into a vein, we actually see improvements after stroke, at least in animal models. So a lot of people thought originally this is just a bystander effect. It's something that is coming out as a secondary or tertiary effect, just like we talked about earlier with what happens with stem cells. So stem cells don't do it by themselves. There must be another effect that's going on. So if you look at all of the research that's sort of been done in this field, there is a lot of different types of things that suggest that there is this homing effect, that these mesenchymal stem cells are particularly good at being able to migrate to a site of injury. So again, thinking about stroke and thinking about how homing might be useful to us, this idea that these mesenchymal stem cells can home in, and maybe they're better than neural stem cells, might actually be important. They also might be able to modulate the immune response. Again, an important consideration when dealing with stroke. So it's not a very efficient process, but we know that they do have this ability to be able to home in. And so people have been trying to figure out better ways to get them targeted to the brain under normal conditions, and then particularly after stroke. And it might turn out to be how you deliver it, where you deliver it is important. So we don't put it into a vein, we put it into an artery, and then it'll get to the brain better than if we inject it into the vein. So the route of delivery might be actually really important, or delivering it directly into the brain using route. But this requires neurosurgery, and we don't necessarily want that. It requires more techniques, etc. So this gets into what actually are some of these extracellular vesicles. And you're going to have to tell me, did you learn about this in your second year bio courses? Has it been a while since you learned this in your second year bio courses? Did you learn it at all in your second year bio courses? I see some nodding, so I'm going to make the assumption that you have. So if you were to take a look, and we start off with exosomes as an extracellular vesicle, or we look at these microvesicles and these apoptotic bodies, they're all a little bit different. And they all have some unique properties that are associated with these different types of extracellular vesicles. So in particular, if we take a look at these exosomes, one of the hallmark features, the thing that makes them different than these microvesicles, are things known as these tetraspanins. And these tetraspanins are labeled for you here. And these tetraspanins are these 4 transmembrane domain proteins. And they seem to be really important in these exosomes, and they help to define these exosomes. If we look at what's inside these exosomes, we find all sorts of interesting things. So we find things like mRNA, we find things like microRNAs, and we also find things like heat shock proteins shown here in purple. And these actually might be modulating how these extracellular vesicles, one class of which is known as the exosome, might be able to help neurons after damage. So again, I don't need you to memorize every last protein that's on here. There are a number, and this list is by no means definitive, but I do want you to know that we tend to find these tetraspanins, and we do tend to find microRNA and mRNA and heat shock proteins within these exosomes. And this is very different than what we would tend to find within these microvesicles, and also very different than what we would find within the apoptotic bodies. And the big one within apoptotic bodies is that we tend to see the fragmentation of DNA and histones associated with apoptotic bodies, but we don't see them in these other categories. So again, we can mark and identify these different extracellular vesicles by knowing a little bit about their composition and what they normally contain. So how might we use all of this knowledge to our benefit? And what could we be doing with them down the road? And why are we kind of talking about all of this? So I've already mentioned to you where these extracellular vesicles, where they come from, actually are really important. So mesenchymal stem cells are different than neural stem cells. And what comes from these extracellular vesicles is also going to be different as well. So it also determines how they might be useful. So certain types of damage, some of these extracellular vesicles might be of more use than other types of extracellular vesicles. But one of the things that we're particularly interested in, some of these extracellular vesicles, particularly things like the exosomes, might actually be able to transfer more than just what's found intracellularly, more than just the heat shock proteins. The cargo contains a number of different factors, microRNAs, mRNAs, et cetera, and we'll talk about that when we get to Alzheimer's disease. But the other thing is we might also be able to transfer receptors from these extracellular vesicles from one cell type to the other. As a result of all the things that they can transfer, it might be able to change whether cells survive or die. It might change how they function. It might even change neuronal plasticity, which is a hypothesis that I'm going to present to you at the beginning of Wednesday's lecture, depending on how far I get here today. And of course, immune responses are a big part of this modulation as well. So these microvesicles and this sort of pathway that you see here are a little different than how we generate these exosomes. So microvesicles normally are bud off from the surface of a cell versus exosomes, which are actually part of the endocytic process. So if you were to take a look and you look very carefully at that diagram that I showed you a few slides ago, you'd actually see more features with things like clathrin, as well as flotilin and other molecules in these different exosomes. And they are derived from these early endosomes, which then form these multivesicular bodies before they get released as these smaller exosomes. So this process, by the way, if you were to take a look at last year's final exam, is one that I asked students to make sure that they knew. It's probably a good idea for you to know the difference between how we generate microvesicles versus exosomes versus apoptotic bodies, which again also include different types of molecules as part of their cargo, which is outlined for you here as well. So these extracellular vesicles are ones that we're going to be spending some more time on. And again, just to go back and repeat several different times, because the data is one that I want you to understand why it was so important, is that we have different populations of these extracellular vesicles. They're all slightly different in terms of size. Microvesicles are a different size than exosomes, which are different in size than apoptotic bodies. They contain all sorts of different proteins and lipid profiles. They contain RNA, DNA, microRNAs, and they're all different in that ability to contain different cargo, and they might be important as a result of communicating between different cell types. And in the CNS, more than any of the other organ systems, because they're so compact and they're so close together, this actually might be the fourth type of communication that neurons actually use to be able to gain some functional changes to the neurons overall. And there is evidence all the way back in 2007 that this pathway might be used to alter synaptic plasticity. So it's not necessarily a new theory, but it's a theory that has really started to take off in the last decade or so. Now this is a really busy slide, and I don't expect you to know all of the details on this slide, but it does kind of fill in some of the gaps that I've been mentioning in those previous slides that I have been showing you. So if we were to take a look, this is the brain, this is the blood-brain barrier, this is the blood in red, and we've injected in these mesenchymal stem cells, and these mesenchymal stem cells that are here are releasing all sorts of these different extracellular vesicles of different sizes, microvesicles, exosomes, etc. And they could contribute to a number of different things. They can actually cause synaptic plasticity. They could cause regrowth. They could suppress immune responses, etc. But if you take a look, the communication that goes on between all of the different cell types within the brain is now also thought to involve the transfer in both directions of these different extracellular vesicles, either from neurons to astrocytes or from astrocytes to neurons, or from the oligodendrocytes, etc., or microglial cells. All of this cell-cell communication seems to be incredibly important right now. So it'd be one of those interesting experiments, if we could ever figure it out, is to block all of these different types of extracellular vesicles and all their communication. What would happen to your brain? We don't have that technology right now. It's kind of beyond our ability to be able to control it, but it'd be really interesting to see what the overall net contribution of all of this type of communication is. So I'm going to take you through one of these papers. This is an earlier 2015 paper. If you want to go in and look at all of the details, you can go and click on this link and it will take you to this paper. So if we were to take and to examine the same sorts of ideas that we looked at earlier, so going all the way back, I know it's been about an hour and a half-ish, but at the very beginning, at the outset of today's discussion, I had mentioned that, we ultimately at the end, we know that neurogenesis and we know that the migration is actually really useful to reduce the size of the infarct, and we also know that functionally it's really useful to us, but neurogenesis by itself is something that's still a mystery. So one of the things that was done in this particular paper is to address some of those gaps within our knowledge. So we know about BRDU. It's a label for dividing, proliferating cells. We can go in and see the incorporation of BRDU in any cell that is dividing, and the more BRDU that we have, the more proliferation that we have overall. So if we were to take a look inside the tissue of an animal that just under control conditions, or after the introduction of the mesenchymal stem cell line 1, so this is one cell line from mesenchymal cells, or we have a mesenchymal cell line number 2, a different mesenchymal stem cell line, still mesenchymal in origin, and we look to see, does the delivery of mesenchymal stem cells enhance proliferation? You can see that it does, maybe not surprisingly. But if we were to derive the extracellular vesicles, we're no longer injecting in the mesenchymal stem cells, but just their byproducts, just what they are normally shedding, as I showed you in this previous diagram. If we're only looking at the byproducts, the extracellular vesicles, how good is that effect? And it turns out that just the extracellular vesicles and the delivery of the extracellular vesicles also enhances proliferation to the same level. Kind of makes you go, makes you think a little bit. Maybe that's the more important factor. It's not the stem cell itself, but what's released from the stem cell. So let's continue on. Let's take a look at the model that we've been looking at. Let's look at what happens after stroke. And we know that there's normally a proliferation after stroke, etc. So let's take a look at double cortin, which is a molecule that you are intimately aware of, familiar with now, and if not now, you will be by the final exam. So we know that double cortin is a marker for dividing and migrating neurons that will go to an area after stroke. So what happens if we inject in mesenchymal stem cells? And does that compare to control? So this is baseline stroke. This is an animal that has stroke. Do we enhance more proliferation and migration after stroke, which is the black bar here known as control? So we do the same two stem cell lines, the mesenchymal stem cells, and we see, surprisingly, we get an enhancement of the migration, that double cortin moving to the area after stroke, and then we also see that both stem cell lines actually do the same thing. But let's take some of the byproducts, what's actually shed from the surface of these different stem cell lines, and not put in the stem cells directly, but just their extracellular vesicles. Do we see an enhancement? But again, strange, odd, curious that we actually see the same size effect, not the cells itself, but the extracellular vesicles from both cell lines actually produce just the same level of effect that we saw earlier. And we could go on and take a look at every last thing, but at this point, you should be familiar with some of these markers. Double cortin is a marker of proliferating cells and the ones that are migrating. So I understand that these stem cells and the extracellular vesicles actually enhance proliferation as well as the migration. And I know what nu-N is. And nu-N is a marker for final neurons. Once they reach the target, they will differentiate into neurons, and then we can see them as these nu-N positive cells. And in fact, if we take a look, we see that the overall effect of these extracellular vesicles derived from these stem cell lines is actually pretty close to what the stem cell delivery is by itself. Curious. Right, so data after data actually suggests that this is really good. The only piece of data that you won't be able to tell me about directly, because I haven't covered it here today, CD31. What is CD31? What is it a marker for? Or if you can't tell me that because you haven't learned it in another class, if you were to just take a look at these overall images here where it's being marked with CD31, an antibody to this protein, what do you think it looks like in the brain? And it's kind of hidden and it's kind of buried in an earlier discussion, but if you're taking a look and you've taken neuroanatomy classes or any anatomy classes, you might be able to make a pretty good guess as to what these things within the brain are. It's a good guess. Thank you. Good voice. You should actually lecture. Not microglia. Anything else? Yes. Not ventricles. Oligodendrocytes. Sure. What else do you have in the brain outside of cells? What else? What do we block when we have a thrombus? We have blood vessels, and these are markers for the blood vessels. And one of the things that we see is that when we look at blood vessel expression using CD31 as a marker for the endothelial cells that normally make up the lining of the blood vessels, that we see an enhancement of the blood vessels after using either stem cells or after we use the extracellular vesicles that come from these stem cells at about the same level. Again, the statistics are terrible. They're not something that we would love to see on a routine basis, but it's very clear that overall the effect is pretty much the same. And so what this suggests is that these extracellular vesicles are good at proliferation, they're good at forming new neurons above what we would normally expect, and we don't need stem cells, we only need these extracellular vesicles. The big challenge, though, is to know what's inside these different extracellular vesicles and how is it causing increased angiogenesis or the development of new blood vessels after stroke and what they might be containing. You might be thinking VEGF because VEGF would be the logical choice, but we'll talk about that in the next lecture. And we will talk about what happens in this model, and then we'll transition into Alzheimer's disease, and then we'll talk a little bit about exosome therapy and other things in the next lecture as well. So for those of you who are still interested in coming in to look at term test 2, at this point it's probably best to just make an appointment with Katie, work out like your timetable with her, and then figure out a time that works for both of you, and then she'll have some viewings in the office. Good luck on any term tests you have. I'll see you all on Wednesday. So how did your TEDx thing go? I saw some live feeds on my Facebook, and it looked really good.
Yeah.
I didn't know that one of my colleagues, Anna, is